before I get started, can you hear me all the way back, or do I have to wear the microphone? No. Okay, great. I don't like those microphones. I don't know if that means, do I need to wear it for streaming? No.
Yes, it will help.
Now we will establish that I don't like those.
Sorry.
Okay. Can you hear me streaming? Good. So, computational animal ecology. I'm pretty sure that at least two of those words are familiar. And in the third one, what is ecology, and what is in general, why would I put computation and ecology together? So here's why. Here's my view of the world of science, and that's very much sort of Poincar's view. As scientists typically have a question that they want to know the answer. For example, why is the sky blue, or why are animals social? Why would we all share resources and be nice to each other and tolerate the density that is Manhattan to be together near each other, right? To be in groups. So when a question is asked, a scientist would like to go through some process and come to an answer which starts with because, right? If we ask a question why, and most scientific questions are why, then we'd like to have an answer that starts with because. But what typically happens is that the question is instantiated based on the experience, the reality of the world, the background of the scientist, the particular version of that question, and we get data. And then, after staring at those data and a lot of intuition and knowledge about the background world and data analysis, there are a couple of hypotheses that come out of this process, and this may iterate a couple of times, many times, and after experimental analysis, we come to we come to a supported hypothesis that provides plausible explanation. And so it's not quite the answer that starts with because, but that's the best current standing scientifically supported answer that may answer the question. So where does computation come into this process? Well, in my opinion, at 4 points. And the most sort of important one, which opened the door to computation in the science other than the Excel spreadsheets in the analysis part and word processing, is data. And there's computational processes and data collection techniques, as well as the technology beyond computation, such as GPS, genomics, all kinds of sensors other than GPS, photo, video, crowdsourcing, and methods for data integration and storage allowed for much bigger and more different diverse sources of data for this process, for scientific process. In fact, so much more different and diverse that today the questions that are asked can be different. The answers that we're getting can be very, very different because we stop looking at the whole world of science through this sort of tiny peephole and open the huge floods, the huge sort of observatory scale data sources, view of science. And so that much data requires very different types of analysis. You cannot just stare at those data to get your intuition because you can't notice the patterns. Patterns is noticing intuition. Intuition is noticing patterns, right? And humans are very good at seeing patterns, even when they're unknown. We can see patterns in random data. We can see patterns in white noise on TV screen. Anybody remember white noise? Right. We can see patterns in the stars, right? We extracting all kinds of shapes and figures. So humans are particularly good at seeing patterns. We're also particularly good at seeing particular kinds of patterns, face shape, for example. We have hardwired wiring in our brain to see patterns in the form of faces. We're also very good at seeing local patterns, but we're not good at seeing statistically significant patterns that are correct in terms of the statistics, not overestimating, not underestimating. We're bad at seeing global scale patterns that occur over long periods of time, but very infrequently. We tend to underestimate the probability of certain events that are sort of long periods of time and overestimate the outlier probabilities. So all of this is kind of based on our own evolutionary experience, but computers are very good at all of that, right? Extracting statistically significant patterns that are well supported in data. The problem is that computational processes produce many of those statistically significant patterns that are not interesting, right? So for example, there's light when the sun comes up. Yeah, it's very statistically significant pattern, but it's absolutely not interesting and not important. Well, it's very important. So what we want is patterns that are important and interesting and statistically supported, right? From data. And so there's a whole question of how we do it, and that's what data mining as a discipline is about, is extracting those patterns. The other big difference between the computational approach to data analysis and scientific is that scientists want explanatory models that produce answers that start with because to questions that start with why, right? That explain why this phenomenon is happening. Well, what computational processes, computational analysis is incapable of producing this. For explanatory modeling, we need expertise in the domain. What computational approaches are very good at, and what we as data miners produce, data analysts, is descriptive and exploratory, descriptive and predictive modeling. So let's get on with it. And why would we want to do that? And of course, visualization and model testing. So I'll talk mostly today about those three sort of boxes: data, analysis, and the resulting hypothesis models. So what is ecology? Ecology is the science of connections between the living organisms and their environment, among the living organisms and their environment, in the evolutionary context, right? So, in terms of, for example, for behavioral ecology, the questions are: why would animals do what they do in the context of fitness, right? Evolutionary fitness, and to sort of facilitate answers in that space with those three boxes in mind, we're building an ecological information system, which that's the data part. So it takes the data, all kinds of sources of data, from geographic information system and genomics to pictures and GPS colors and so on, and so it integrates it into some database. And we have analysis part to it. Everything from counts of population, what's the population size, what are the home ranges, what are the people are interested in about animals. Animal ecologists are interested in the question of cognition and behavior in terms of fitness and survival, population dynamics, invasion and dispersal of home ranges, and social behavior. So we'll focus mostly on the question of why are animals social, and try to answer that question. And of course, I'll come with models and computational tools which produce statistically significant predictive and explanatory hypotheses, testable hypotheses. But we also need visualization, and a nice byproduct, if we can do that, is education and outreach. Okay. So traditionally, here's how ecological data were collected. If you do search in any of your search engines on ecological data collection, this is pretty much the images that you get. One or two people, sometimes a few more, sitting in the field collecting field data collection, right? Collecting data from animals, from grass, observing animals, writing things down on a piece of paper, nice 15th century technology. And occasionally having some sensors. So there's a GPS sensor maybe, and light sensors, and maybe a camera to take pictures, then sometimes going to the lab. In fact, this is my postdoc, David Papano, who is collecting data on social behavior of geladas in Ethiopia. That's, so up until a few years ago and still today, typically social interaction data is collected by standing there, observing your population, so geladas really don't care that a human is there, and writing down in two ways. Either you follow a focal individual and notice all the interaction that that particular individual has, or you do scans of the population and write down every interaction that the two individuals have. Okay? So clearly that's not big data, that's not a lot of data to analyze computation, and most of it can be even analyzed by hand, or in Excel for sure. So here's how today data on social interactions are starting to be collected. So this is from Switzerland, from University of Lausanne. These ants are tagged with QR codes and followed by cameras, so the camera can follow the entire colony in this case, the entire nest, and there are 30-something nests of these ants, and they track every interaction, every ant, every location, and they have a big system there. This is a different way to track ants from Arizona. They color code them by spots, and then you can sort of uniquely identify, or you can write down a number on each bumblebee. Well, great jobs for graduate students, right? Okay, well, that's in the lab. Wonderful. What can we do in the field and with bigger animals? Well, you can put GPS collar on each zebra. There it is, with a solar panel. It takes about $3,000 to build a GPS collar for an animal in the field. It has to be with a battery that lasts for a while. You have to get that to tranquilize each animal before you put a GPS collar on it. So it's a big operation. So suppose you could do that and put data and put a collar on each animal. You can track them and then have a sufficiently accurate location for each one of those. And now you can build a network of proximity. If the two animals are close together, then they must be interacting. The problem is that not always, of course, they are interacting. They're not even facing each other. So it's fine. You presumably could eliminate those edges by orientation, direction. So there's work done by a biologist from formerly of Princeton, now officially from the University of Constance, Max Planck Institute in Germany. This is Grand Central in New York. Video just automatically identifying the gaze of each person, so you can now extract interactions by matching the gaze direction. Great. So this is a project of which actually Ivan Berger, who is my current PhD student sitting here, is part of collaboration with Dr. Matt Crawford from the University of California, Davis, tracking baboons, so putting GPS collars on baboons. They tried a troop for 30 days. Each we took GPS location every second, so 1 hertz resolution. And you can extract this proximity network. So here's 1 day in the life of baboons. And Ivan can tell you a lot more about extracting latent networks from those and other data, so watch that space of extracting latent networks from data. So yeah, watch that one guy, right? Running back and forth. Come on, let's go. Yeah. So we'll come back to baboons and these other animals. But even by putting the network, look how much information and network structure gives us. Just by drawing that and visualizing, I haven't shown any analysis yet, you're already starting to have hypotheses about what's going on there, right? Maybe there are 2 groups, maybe there are 2 types of baboons, 2 subpopulations, who knows? Right. So there's also a project that's about to be launched in January 2015 in Germany, led by Max Planck Institute and the University of Constance. Martin Bikatsky, Icarus, the International Collaboration for Animal Research using space. So that's a pretty amazing way to track animals using the International Space Station. So here the size of animals you can track using that. They'll be able to track insects. Here's a little receiver transmitter that is passive active. It's activated by the space station antenna, and data are collected. So you can do birds migration and so on. No. And butterfly migration. But you have to put a sensor on each animal. So all of this so far requires putting a sensor on an animal, which is very, very labor-intensive process, but you get a lot of data. What if you didn't? What if you now switched all your resources towards not putting everything on animals, but instead getting a lot of information about the animals through technology? So video, as we pointed out, if you can identify animals, like QR codes. So we can use drones on horses or on zebras if we can identify each individual zebra. And in fact, we can, and I'll tell you in a second of how, but each zebra is unique. They have patterns on their stripes. And if you can have a drone flying over it, you can get videos now and like just those ants tracked. We tested that in Kenya. We work mostly in Kenya, so this is Sarah Vela and Dan Rubenstein, the main collaborators in this. And this is a graduate student because we couldn't get zebras when the drones were flying for the pretest. So that's what graduate students do in this space. Michael Brown. So you can also get pictures of animals by putting stationary motion-activated cameras. So this is there is a camera trap, 2 of them, and that's a project from a field computational ecology course that I co-teach with Dan Rubenstein from Princeton, ecologist. This is from 2012. Bob Poise, who is a computer scientist, who is a biologist in this. So this is a course which brings together students from ecology and computer science who give background in both disciplines, and then they work in interdisciplinary projects. And so here's they're working in an interdisciplinary project, computer scientists, biologists, and natural resources master student. So you can also get pictures from tourists or field scientists who are going out in the field with cameras, photographing animals. So all of this is great if you can identify individual animals from these photographs somehow fast enough, automatically. And that project on identifying zebras from photographs by saying this is Kathy, this is Joe, this is Paul the zebra, was one of the first projects in the field computational biology course in 2010 when we first taught it by Mayan Tahiri. So we got popular press. The height of our achievement was NPR, Wait, Don't Tell Me, but also National Geographic and everywhere else. There's tons of data now coming from handheld devices, citizen science projects, uploaded Flickr streams and Google Plus and all the other photographs and Instagram, people taking pictures of animals. But there's also animal species identification projects, like Snapshot Serengeti, Smithsonian Wild. There's Wild Me, we work with the nonprofit Wild Me that takes pictures of animals such as sharks and whales, and you can upload those. So there's many platforms now that bring in images of animals at large scale. And if so, then we need a system that can take in, ingest all these pictures, and produce usable scientific data for analysis that then results in usable scientific insights. So this is a project we're now focusing on, the particular version of the ecological information system, image-based ecological information system, IVIS. There's our beautiful new logo. And it's a project in collaboration with Chuck Stewart from Rensselaer Polytechnical Institute, Dan Rubenstein from Princeton, and Jason Holmberg from Wild Me. Guess which one is actually the biologist? No, actually, this is the Dr. Who looking one. So that's because both sides are true. Right, Dan Rubenstein is an ecologist from Princeton, and everybody else is a computer scientist. So this ecological information system, the input comes in forms of images. And so all of those sources of images that I mentioned are only usable if we can identify individual animals. And so that part was developed in collaboration with Chuck Stewart from Rensselaer Polytechnical and his student, John Kroll, the basic program is called pot spotter. We can identify now anything that's striped, spotted, wrinkled, a notch. So we can say not only this is a zebra, this is a giraffe, but we can say this is Cat the zebra, Joe the giraffe, Paul the elephant, Jerry the jaguar, and so actually this is, sorry, yeah, it is jaguar. Leopard, sorry. Leopard or snow leopard, even hippos, toads, frogs, rhinos. There's a story behind this picture. We got that close to that rhino. And nautilus and fish and many other animals. And there's the sharks. So we can identify those. And the elephants are by wrinkles, by the way, so it's like fingerprint wrinkles. They're all unique, and they're all pretty easily identifiable from any angle. So now we can take the pictures in thousands and re-identify animals and track them without putting GPS collars. So this is massive data. But images, while great, wonderful, plentiful, free, and exciting source of data about animals, they're also hugely biased. Like any other data, scientists standing there with a pen and paper writing down all the interactions about animals is also hugely biased. It's biased by whether the scientist is there or not. It's biased by what he or she can observe, observer bias, what they interpret as an interaction, and so on and so forth. We've tested all of that. There are plenty of papers documenting the bias of the resulting data, even though pretty much everybody would like to ignore it and think that scientists collect perfect, expert, objective data. But images have particular additional sources of bias. So camera traps are biased by the location of the camera. It's a stationary location, so the animals have to come to it, right? The sensitivity of the motion sensors, so they have infrared beam, which is when broken, it takes a series of 3 pictures. Those are the motion-activated cameras. And this beam is broken ideally by an animal moving, but it also can be broken by grass, by moving, by all the branches around, right? No matter, if you're going to clear the space around the camera from actual nature, you're not going to get the animals that live in that habitat. So you have to leave this space around more or less untouched, and so then you have all the nature moving, other than the animals. It can be as sensitive as, if you put it too sensitive, then it can be activated by the contrast of the shadows as well. So then you have to deal with lots and lots and lots of false positives in the pictures. In fact, it can be more than 90% of false positives, right? So then you have to remove it. And background removal doesn't work very well for these cameras, for these pictures, because they're only activated when something is happening, so you don't have the true negatives. And after working a lot and trying to do background removal from these pictures and using all kinds of algorithms and training them on features and trying to identify the ones that contain animals, turns out the easiest thing to do is not to create better and better algorithms for animal detection in those pictures, but to change the protocol on the camera traps themselves. And instead of just being motion-activated cameras and taking a picture when the beam is broken, in addition to that, they should take a picture, let's say, every hour. So we have a baseline. And then background removal, the simplest background removal algorithms work. And so we're working with Roland Case, who is the curator of most of the camera trap pictures for the Smithsonian Institute, which is sponsoring a lot of this research out there in the world. So with this, we're talking about more than, several millions of camera traps throughout the world. That will be then changed with this protocol. Okay, there's also the quality of captured image of an animal. Sometimes you just get a tail, sometimes an ear, sometimes the back of the head, and sometimes you get the whole animal. Great. So drones and other cameras on vehicles, either flying or underwater or driving land vehicles, are biased by the geometry of the route that they take, by the presence at a location when the animals are there, and the resolution of the images. So how high should they fly, with what camera resolution, to be able to identify individual animal. right? So we can get away with about maybe 200 by 200 pixels for identifying zebras, but what if they have smaller spots and so on? Yes.
And a quick, so with the bias with the images, how about weather? Does weather also weather bias?
Yeah, visibility, so that's the quality of the captured animal, and so here as well. Weather, in this case, it's a good point because drones cannot even fly in high winds. We've lost two. But they're easily refurbished.
Okay. Okay.
But it was a spectacular crash. But it's a styrofoam drone, so you kind of put it together, glue it, duct tape it in. Okay. And so the human observers with cameras are biased by many, many more things than that. Of course, there's the observer photo selection bias. So what do I decide to take a picture of when I see it? And right now we're working at a nature preserve in Kenya, Old Pajeta Conservancy, where tourists are taking pictures. We're working with the tour operators. And we have a field assistant, Michogu, who is driving with the tourists and noting down every animal that is there every time that stop versus, and so then we can compare what the tourists could have taken pictures versus what they are taking pictures of. So we have the baseline from hopefully many tourists in the future. The goal alignment there, the tourists, for example, are there to see the animals, and the more experienced tourists, I'll show you in a little bit, are there to see exciting pictures of the animals, such as a lion taking down a zebra, or wild dogs taking down an impala, right? Everybody, for some reason, is interested in these warrior versions of nature. Or animals having sex. So, but nobody, after you see about 100 zebras, nobody wants to take any more pictures of zebras. Which is the species fatigue. There is also the charismatics of the species, if they're doing something exciting, if the species are interesting. So all of that, there's the range of training and expertise, there is the demographics, and there is well-documented, so here's where also the diversity comes in. We need the diverse sources of those data because there is well-documented biases by sex, age, country of origin, and so on and so forth. So here's a little bit of preliminary data. We have, this is on one tour, there are 4 cars, about 4 or 5 people in each car. So this is all people that went on the same tour during the same 2 hours. The number of pictures that they took ranged from over 450 to less than 10. Right? So how do you do mark recapture statistics from that? You have to know these distributions. Here's the rate of pictures per hour per hour of tour from experienced versus novice. This is combined from 9 different tours. Of the experienced tourists were Earthshare volunteers. This is their 5th, 6th trip to Africa versus the novices. This is their first trip to Africa, their first safari. So on the first hour, they're excited. They take lots of pictures. By the second hour, they take fewer than the experienced tourists, who take pretty much the pictures at the same rate. They know what they're getting. They know what to expect. So all of these, we're now collecting this background distribution so we can create these statistics, which, because all of this results in biases for species, which species are observed, which is not, are observed in the data versus actually present in the wild, which individuals are population count. So if you want to do market capture statistics, that's how you count population. So you, if you saw, let's say, 100 animals today, of 100 zebras, you wrote down all of their identities. You come tomorrow, you see about 100 zebra. 10 of them are the same ones you saw yesterday, so your recapture rate is about 10%, which means that if you see 100, the population size is about 1,000, right? So that's the very simple version of it. There are very complicated market capture statistics. If you want to do that from pictures, site to site, you need to know your biases. Spatial distributions, all of this is bias of spatial distribution. So if you do home ranges or track individuals or species distribution, it's all, you need to know all these background biases and the distributions of biases. For Beth events, interaction, which is social behavior, rare behavior is hugely biased, endangered species and cryptic species, not easily identified. The data on those are very biased. And so, which the sample queries that we would like to know is the first and basic is population count. You cannot do any ecology, population ecology, you cannot do any biology at the population level if you don't know how many individuals you have there. Right? You cannot do conservation or population management of endangered species if you don't know how many individuals there are. And so this is an example of hotspot, or not the entire system yet, deployed in Nepal, working with some ally scientists there, who works with school kids who take camera traps and the nature preserve, the snow leopard conservancy in the Himalayas. And these are pictures of snow leopards from camera traps. And our system has provided the first, is in the process of providing, because we need to verify from 2 years at least, the first population count of snow leopards in that area. And actually there are more than people think there are, but we're not disclosing the numbers yet because they're highly endangered and we need another year of verification. Also, the system was used to count the gray zebras in Kenya. And the news there are actually bad because there's about half the number of this highly endangered species in the area that everybody thought there were. Instead of about 400, there's only 220 with many, many fewer juveniles. So this is of a species. Was considered about 10% of the species being present there. So that's how endangered those species are, those individuals are. And so if we have half the number than everybody thought there were, that's bad news. But we need to know that. And here's a more complicated query. This is an ecologist challenge, and then essentially once you can read the whole thing, but they sent what Blair asked us, Blair Roberts, is the survival rate. Can you give me the survival rate of juveniles, of giraffes, from this data? And so what it would translate into, can you, for every giraffe mom, can you tell me who the offsprings are when they were born from the photographs, or rather when they were first sighted, and how often are they sighted after that, and when they stop being sighted, right, in the images. So all of that we can do if the sighting rate for juveniles is unbiased, or if we can correct for the bias. So all of this is in the question of how do you correct for the bias. And what we do, one of the first things is to realize that each individual photograph is not representative of what's going on in the wild, right? Because we photograph a zebra or 2 zebras. Or we try to focus on fighting zebras or drinking zebras, or maybe mom and baby zebras. But this is a picture of one population, one group of zebras by the waterhole. So we somehow have to combine them into this notion of encounter. Here's what comes out of very quick combination of the previous photographs, which gives you, and now we're actually building much better tools for creating this synthesis, and I can show you big panoramas of about 500 zebras that sort of in one shot together. So that's the basis for our not individual photograph, but sort of collection of these photographs encounters that provide the basis for the statistics. And so we have the initial system based on this encounter idea from Hotspotter identifying each individual animal and working with WildMe database for animal sightings. So we've deployed the system in Kenya at Ecology Nature Preserve. This is training the 2 field assistants who are working there on the zebra database. This is a demo and about to start the project in Leva Conservancy, also in Kenya, that is the Nature Conservancy's own Nature Conservancy. And they work on elephants mostly, with elephants. Okay, so the guts of the database is Wildbook. Wildbook was initially created for sharks and whale shark sightings, and can take all kinds of data, including image data, if it can be processed and animals can be identified. Until now, it's been mostly by hand. Then they were switched to the software, which was kind of slow and didn't scale up, but we will. And it can work with all kinds of statistical programs, including mark recapture, where it's marked somewhere. There's a program mark, and all kinds of other statistical analysis and other data such as Google Earth and GIS and so on. So, but the guts of it is holding data on animal sightings and biology of animals in a standard which is Darwin Core compliant, which is a very important thing that you can export and exchange data. Okay, I'll show what's behind there is you can also export that data to social media, and I'll show a picture in a minute. So you can now adopt an animal. Once you go to a safari, so what the experience is going to be, you're going to a park, you see an animal, you photograph an animal, it tells you, oh, that zebra you just saw, here's all its information, and would you like to adopt it or follow it on Facebook? Or, and on Facebook, you'll see all its friends that it teams up with, and so on and so forth, from photographs. So if you can to do that, we need to go back to social networks of zebras and other animals. And how do we extract that network from photographs? So watch that space, watch Ivan, he's going to be his PhD work. But then suppose you can extract that information in those networks. So the question is, with all these biases, is there any way of doing it in a principled way if you know that the information you want out of it is network information? And so creating these biased creating these samples of networks that give you useful information was the work of my former PhD student, Arun Maya. So the idea is for network analysis particularly, you have a sampling strategy which produces the properties of your sample, and you can then infer the properties of the network. And everybody was complaining about sampling bias, we too, right? So I just spent about 10 minutes talking about sampling bias. And everybody was trying to correct for the sampling bias of networks specifically. But the thing is, and that's Arun's idea, that don't correct for sampling bias, use it if you know what it is. In fact, sampling bias is an asset to be exploited similar to the concept of stratified sampling, if you can control how biased you are. And so here's the idea. Typically, how do we sample networks? Well, the simplest way, which we're all taught sort of at the beginning of our computer science career, is breadth first search, depth first search, maybe random walks. And there's been work that showed that essentially the probability that you see a particular node in the network is proportional to the degree of that node. So that's bias. Then forest fire sampling approach, which is essentially probabilistic breadth first search, and has similar bias. Then there came degree-based sampling by Lada Damage et al. And that's, you take a node, so all of these are iterative sample strategies. You have your current sample, and then you're asking their link-based traced sampling strategy, link-based traced. So your next addition to the current sample is based on the nodes, is from the nodes that are connected to the current sample by links. Great, so which node do you select? Degree-based sampling tells me take the node with the highest degree. Okay, all of them have the same degree. You can propose more. We propose sample edge count induced subgraph. So take the node that has the highest proportion of edges pointing in versus out. You can do the expansion, which is the reverse. Take the node that's the highest proportion of edges pointing out. So I'm sure within 5 minutes of talking, we can come up with about a dozen more sampling strategies. That's not the point. The point is, what does it give you? And what we want is a representative sample. Meaning that if we look at the entire network and are interested in something, let's say, the community structure, everybody's favorite question for some reason, community structure of that network. So here's the yellow is the entire network. And I run my favorite community identification algorithm, which gives me these 3 communities. Then what I want from my sample is the following. The blue is the sample. I want the sample that if I take it and run the same community identification algorithm on it, I will come up with the same structure as if I took the whole network, my community structure there, and restricted it to the sample. Okay, so that's a representative sample. And you can do it for any question other than community identification. So essentially, the sample that we want is look ahead. What is it that the question that you want to answer, and create the best sample that fits that question. And you can analyze all these sampling strategies that we showed, that we suggested, and whatever your favorite one is, And you can, in some cases, prove that for some question, the particular strategy is the optimal one. So, for example, expansion-based sampling is optimal for community structure and for degree for nodes with the highest degree, for producing nodes. Sorry, for producing in the degree count structures are best for nodes with the highest degree. These ones are, even though they're biased by degree, they're not as good typically in experimental analysis. Okay, the next question, if you're going to sample a network which changes over time, is what's a good time step? And that's a question that was started to be answered by, again, my former PhD student, Raimonda Caceres. And the problem is with dynamic networks, if you sample them too frequently, such as every second of the baboon network, or every second of email network, right, then what you're going to see is either very, very sparse networks or networks that don't change very much. Baboons don't move enough in 1 second to really change the configuration of the network. If you aggregate them over too long periods of time, so if you aggregate the entire day of a baboon in one network, you're going to lose the important information about the order and the actual causation of interactions, right? So somewhere between the very, very frequent sampling and the aggregation over long periods of time is the just right sort of time slice, which somehow maybe corresponds to the temporal scale of that network. And for human contacts, even though the title of this paper by, this is from Cecilia and Marcolo from, Cambridge University. The title is on non-stationarity. It actually turns out that there is stationarity in human contact networks. It's just a different scale. So there's your days. Sorry, this is in the terms of hours. So you have scales, right? So this is about daily. So every half a day there is the day-night. There is the day after day periodicity. There is the week after week. So there is weekends and days and so on and so forth. So you can kind of see that there is a rhythm. There are scales in this time in this network that correspond to something real. And Raimonda's work showed that if there exists one scale, stationary scale, you can extract it. If it exists in the latent data, it exists. You can extract it in the actual network data. And she provided an algorithm, and I'll just sort of what it does when you extract the real scale, the good scale, then things like rare events, the change of stationarity, and the periodicity come out in the sort of good scale, temporal scale. Okay, so we hopefully can get the biases of data. We can sample it with the right biases. We can sample the right data, extract all of that information from images and other data. And now we're maybe starting to getting to the point where we can answer, so why are animals social? And one of the biggest tools, one of the best tools turns out, is to look at the persistence of social interactions in animals. And that means communities, sort of groupings that are kind of persisting over time, even though they change maybe a little bit. So this dynamic communities, and that was work with my, again, former PhD student, Tanya, and I was in sabbatical last year, so most of my PhD students are former. Oh, I'm here. So if you have a stack of observations of your population over time, you want to somehow say, okay, well, I see these groups at this time step, and then they somehow change. And so what I want out of it is say, well, okay, even though they change, there are these 3 communities, and there's maybe there are core members of the community, and then there are individuals that maybe switch affiliations. This was the number 2 was member of red community, but then it joined the green community. 3 was red, but it just visits the green community. So we want to count somehow for these events of switching community identity and visiting it, because those are 2 real biological events, including in humans. And so there is enough work on community in general, and it typically is defined as a cluster, right? Community is a group of individuals who are relatively closer, more frequent interactions somehow to each other than they are to individuals in other communities. And that's a social sciences definition. And as such, it is a definition of a cluster, right? And so recently there has been enough work on, there has been body of work, accumulating body of work with dynamic communities, all of them either tracking communities or again defining it in some form of cluster. So we also define it as a form of cluster based on a social space, latent social space, defined by costs on certain social actions with respect to communities. So individuals, we posit, based on a lot of biological, social sciences evidence, that if individuals are indeed, if there is such thing as communities, and what we observe through interactions is somehow, based on this community structure, latent community structure, then individuals are reluctant to switch community affiliations, but when they do, there is a cost. And typically the cost is observed in terms of the increased stress, stress hormones go up, so cortisol goes up, it's measured. Harassment levels go up, access to resources goes down, so why are we social, even though we share resources? And social status drops. You can also visit a different community, but you pay a cost. And the visiting cost is also measurable, but it may be different, and the ratio can be either way between the visit and switch. Sometimes if you're visiting from a different baboon troop, you are going to be harassed more than the member of your own baboon troop because you're considered to be attacking. Sometimes you're just a nice guest, right? And so there's a cost of absence, which is measured in loss of social opportunity. Okay, and we can formulate it as an optimization problem. Find me a community structure given these observations that minimizes the cost. You can also prove that this is maximum likelihood explanation for a particular model of community structure. There's a lot of nice theory behind it. I'll skip the model. I'll get back to straight to the results. Here's the baboons. I haven't did a lot of that data, so replicate a lot of the data. So baboons, we asked the problem with inferring the latent network of baboons, there is no ground truth. We can't go to baboons and ask them, are you friends, guys? Okay, so we can extract a proximity network, right? If you're close enough, for long enough, we'll consider you a link in the network. What does it mean to be close enough, long enough? You can impose all kinds of thresholds, right? But they're arbitrary. So it would be nice to have a ground truth, and for that we actually need to go to biologists and ask them, so what's a baboon friendship looks like? When is the context when baboons are really sort of the proximity corresponds to social interactions? And about 3 baboonologists unanimously told us it's when they sleep. The sleeping network doesn't change. They have to trust each other, to be next to each other while they're asleep, so that's the real social network. Okay, no problem. Let's analyze their sleeping network. So here's an hour before wake up. They sleep on trees, so here 2 baboons, here's some baboons, here's some baboons, right? So if you put the threshold small enough, right, when they sleep, so we said, okay, less than 10 feet for at least, I think 10 minutes in this case. That's our definition of an edge. So this shouldn't change. Everybody was told, all people who study baboons asked 3 different labs, 3 different groups, studying different baboon troops. They said the same thing, shouldn't change, night after night. Well, here it is, the dynamic communities of that sleeping network. And you can see quite a bit of community change there. There's a lot of membership change. And in fact, if you zoom in a little bit, there's a period right here where each red edge means a switch of identity, of community identity. There is like almost completely arrangement at this particular moment of their sleeping groups. And so we asked them, okay, what was going here? We asked our baboon expert, Dr. Crawford, who collected the data. So, and she looked back in the data and said, oh, there was a leopard in the area. A leopard is their main predator. So they were actually slept in a different location. Right? So data don't lie, although nature always wins. But So here's a hypothesis now that we suggested. First of all, that sleeping networks may be driven by a different relationship than friendship. And in fact, one of the baboon experts, primatologists, suggested that it's not about social interactions and trust. It's about having the prime spot on a tree, which is as far away from a leopard as you can get. Right? Maybe they sleep on trees that are over the river. So okay, we're suggesting different hypotheses for the dynamics of sleeping arrangement from data. I'll skip the general model of the dynamic communities. Again, lots of wonderful math and theory. You can get, for zebras, this is the endangered species of gray zebras that we recounted. If you look at the sort of network, so they were observed, photographs and human observation, over 9 months, about once or twice per day, fixed loops. So this is like your camera on a UAV, or in this case on a vehicle, with a human behind it. And so here's the resulting dynamic network. If you aggregate it over a whole 9 months, here's your traditional static network. If I asked you to point to a community here, what would you do? Show me a community. That thing, right? Of course it is a community. So I would be asking it. So if you look at the dynamic network, this big blob is composed of mostly this red and green community with a few sort of additions that pop in and out. And if you look a bit closer, the green ones have mostly L's after their identity number, and the red ones have N, and this one's an S. So L is a lactating female, this is the non-lactating female, and a stallion that has to hang out with a lactating female. So it points to some biology behind it of why these communities of efficient fusion species that can split and merge would actually be different communities because they have different resource needs. And if, in fact, if you compute all kinds of statistics on this community structure, such as they do for each individual, you can compute the duration of the community, the size of its community, the heterogeneity, the purity of how many visiting members there are, and so on and so forth. So you get this large, highly dimensional statistical space where each individual now is a point in that space, right? And turns out that even though it's high dimensional, there are two principal components in that space that explain most of the difference between individuals. And so here they're all mapped onto the first two components. There are many points that map to the same place. And so here's the different, the switching faction, how much individual switches, visits, the size of the community, all of these are different parameters. Now, I'll help you. Here's the 4 clusters that are in that space: the non-lactating females, stallions, bachelors, and lactating females. So this high-dimensional space clearly is driven, their social structure is clearly driven by biology, by demographics, right? Which, with the different resource needs and different tolerance for trading resources for something that the social interactions give them. So why is it? Why are they social? And to help answer, we'll look at 2 species that are highly similar in everything else, and until very recently they were considered almost sort of essentially identical in terms of their social interactions. In fact, so this is the same gray zebra, and this is wild asses. And they are both about the same size. They are both equids, horses, but they're efficient fusion social species, so they live in groups that split and merge. They have territorial males and harem females that decide whether to stay at a particular territory of a male or not. And they have about the same physiology. Their ranges overlap quite a bit. And yet, this is plentiful and not endangered, and this is. So what's different? And not that we told something hugely different to biologists, but there is something in their structure, social structure, that is different. But if you look, and it was very qualitative when we talked about it, but if you look at their static network and measure different parameters about their static networks, they're about the same. So if you look at their dynamic networks, however, they look different. Here's the community structure, and even without knowing what it tells you, you already see that they're different. Now, the theory of behavioral ecology and evolutionary biology tells us that there is the trade-off between resources and danger, right? And the animals are social. So in this sort of, there's an envelope. There are the two curves you can imagine as the demand for resources and the availability for resources and the danger. So there's a cost-benefit analysis that's going on. And the animals are social in a particular region of that space. And at the two extremes are the resource-constrained animals. So if they all have to go to the same water hole or the same coffee shop or train station, right? The resource is the constraining factor. Then you'll see animals in groups, because they have to go to one particular resource. It's not that they want to be with each other, but they have to be in groups to get it because the resource is there and it's not enough of that resource. So if that's the reason, the resource constraint is the reason why animals are social, why that species is social, then what you'll see is not a particularly cohesive community structure, not particularly persistent interactions, because they don't really care who else is there. They just need to get the resource. On the other extreme of that is the danger-constrained species, which means that the animals in groups and those species to avoid danger. The extreme example of that is birds that flock and fish that spool, right? They seek strength in numbers to avoid predation. You don't want to be the only fish out there when the sharks are swimming. You don't want to be the only human with your particularly radical opinion screaming it on the corner. You want to have like-minded individuals behind you. And in fact, if you're trying to avoid danger by seeking strength in numbers, you want to have individuals that you trust, that you have some history of interaction, that you know that they will support you, and maybe even sacrifice a bit of their fitness to protect you, to contribute to that strength in numbers, right? So then you do see, for species like that, that aggregated numbers to avoid danger, you'll see indeed a cohesive social structure. So, back to these guys. One of these two has almost no predators, but is severely water constrained for most of its territory. The other one has high predation. So guess which one is which? So what do you think? Resources or danger? Resources, right? There's very little cohesive social structure. The communities that adhere are short-lived and sort of small, and with high perturbation of membership, right? Versus here, despite the fact that they split and merge, it's a very persistent, cohesive community structure. So these guys have lots of lions, and I can show you some very gory pictures. No, it's okay, right? So here's, and that means that visualization is sufficiently good that with little background you can now tell me what the biology behind this. And so here's the beginnings of the answer of why animals social. And in fact, if you take this high-dimensional community social space and put all these individuals, both species, into that space, the separation is actually amazingly clean. You have all the gray zebras versus the onagers, which is wild asses, and the males versus females, sort of in 4 different quadrants of that space, which points to the different biological factors that drive their social interactions. And we can talk more about it. And so the other part of the social interactions and the social network that we're building is with the help of wild meat. So I mentioned that they have once you sort of get all the information and sightings of animals, you can now build so they have a wild meat Facebook app, which I encourage you to join. If you're observing animals and getting pictures of animals, then you'll have, right now it exists for polar bears, sharks, and whales, but we're building it for zebras, giraffes, and anything striped-spotted, wrinkled, and notched. So you can follow your particular animal. You can see who its friends are. So this is right now most popular, but it will be its friends. And so the friends will be extracted automatically from images, who it's observed with. You see who follows it, that's humans. So these friends are animals, and the followers are humans. And so you can build a social network of other zebra lovers or giraffe lovers. The big question for us is what happens when your favorite zebra is eaten by a lion. And you can donate, you can adopt your zebra and contribute to conservation. And this is my particular snapshot of the current state of of my professional social network. And with the generous support of Microsoft, of the Jet Conservancy, National Science Foundation, and the Apollo Research Center in Kenya. Thank you. Yes.
So, you obviously did a lot of image recognition and tracking. How does how do the challenges of doing that on animals differ from the challenges of doing it or do they?
Fantastic question. Thank you. So there are two aspects to it. Part of it is image recognition, and for humans, most of the image recognition, individual recognition, is done through faces recognition. And face recognition software today is very mature, very good, and totally doesn't work on animals. So it's highly, the feature selection today and the whole optimization, the whole classification part of it is very specialized to humans. The second generation face recognition software, with playing with it, maybe with some training, some tweaks, will work for primates, so baboons and other primates, and lions, so forward-facing predators. And but what we do, it's very different. We use very different set of features for animal recognition. So right now it's the variation in their body coloration. So that is the image recognition challenge. But there's also the whole deformability of animal body, right? So when you're talking about a leopard that can be folded in various ways, that's a problem. You have to actually account for this. So there are many challenges in the image part of it, the fact that they're rarely actually posing, and if they're posing, it's probably biased towards that particular location where they're habituated, and you want to get a bigger picture of animal distributions. The other part in general of tracking animals and getting data on animals is the fact that, as I mentioned, that it has to be done quite often. And look, if you want information on wild animals, it has to be done in the wild spaces. Not all of them are easily accessible, and you don't want all of them to be easily accessible. In fact, the snow leopard conservancy is highly restricted. Very, very few people are allowed there. And if they aren't, then what kinds of modes of data collection are you going to get? You can't, so for most animals, you actually can't capture them. You can't put tracking devices on them. If you do, they have to be energy constrained. So battery has to last. They have to be ruggedized, so that it's about dust, water, everything else. They shouldn't be able to be too loose because they can get caught on all kinds of the colors. We lost a zebra due to strangling itself on a tree, on a branch. The collar got caught. Another one, the battery burst in fire, so that's not good. We were able to save that zebra. So all kinds of sort of engineering challenges to actually be able to track those animals. You have for some animals, so with a space station, the devices have to be very, very tiny, but then you have to glue it on an animal with a particular type of glue. For proximity sensors, we have a project in collaboration with Robin Kravitz from University of Urbana-Champaign, developing not GPS sensors that will go to location, but actual proximity sensors, direct proximity sensors. And when we talked about the project to engineers, everybody said, "Oh, that's easy. You can, yeah, I mean, all the Wi-Fi sensors are right, but you're working in areas that are not instrumented. There is no Wi-Fi, right? There are no antenna. You cannot put a fixed antenna and hope that your animals will pass by it." So okay, then they said, "How about, oh, there are these ultrasound sensors, proximity sensors." That's not good because a lot of animals actually sense in the ultrasound spectrum. Just because humans can doesn't mean everybody else can. They're magnetic. The sensors that you use the magnetic field, right? That's again not good because it messes up navigation of most of the birds. In fact, birds, just like we have particular structures in our eye that see the light bright spectrum, the light sensitivity, and translate it into images. Birds have particular structures in their eyes that sense the magnetic field and visualize. So they actually literally see the magnetic field of the Earth and the changes in the magnetic field, and they use a lot of it for navigation. So you can't mess that up. So there are a lot of engineering challenges in tracking animals, and which a lot of them actually I find exciting because that means, there's a real question to be answered if you can solve the engineering problem. Yes.
Yeah. So I was wondering. I think we pretty much related to what you were talking about for tracking animals. How do you actually determine the actual camera sites? So I think some places like you talk about snow like there's like maybe one location or one general area.
Oh, it's a huge area, so you can't just put one camera and hope that all the leopards will parade in front of it.
You got migrating animals that go back and forth depending on season, right? They go travel long distance. You just like eliminate.
So migrating animals, you typically, so the International Space Station, there are a couple of things. Migrating animals, like bird migrations, they're cons and pros. One is that you can't just put one camera and hope that you can get enough data day after day. So you do put sensors. But the whole sort of efforts of tracking migrations over the globe of animals, of birds particularly, is a very mature effort. So Cornell has a big citizen science project of eBird, which is now many years old, where they track birds, migrations in North America, and bird counts as a big citizen science volunteer effort is almost 100-year-old effort both in North America and in Europe. So England, UK has Christmas bird count, where you just count every bird that passes by. And people, bird watchers is the biggest citizen science or volunteer animal watching group in the world, and the oldest one, and people just love there are huge competitions in bird counts and identification of bird species. And now, the same group that does International Space Station Icarus, they're putting little 40-gram cameras on many birds, and so they can actually track migration routes on bigger birds, obviously. So, in fact, some of their, I think geese, Egyptian geese, some geese or cranes, I don't remember, with the bird cameras were captured in Egypt as spies. And this was because they quite often pass through areas that humans typically don't, including, so they had real problems with some of their birds, but also some really good data from Sudan during the whole civil unrest, in fact. But so some of them could be considered spies, I guess. But in general, so migration routes, of birds particularly, migrating birds, is an old effort to track those. We have a pretty good idea of how they go, but it turns out from some preliminary data, not as good as we thought. And they're also they're reasonably fixed, the migration routes. So the Hawk, I think it's Hawk Mountain in US, you can stand there during some particular times of year and just count the birds, all the birds in thousands and millions, in fact. And now they have effort of using the satellite images to count birds. So you can't identify individuals, but you can count the numbers and track the bigger bodies. They show up on satellites. So densities and routes, doing that. Now for insects, it's a different problem. And so how do you do butterflies? And how do you when we know the migration routes, that's the non-interesting part. That's the equivalent of there's light when the sun comes up. What can we find that we don't know from those data? Okay? And there's now, after this has been in place, not the space station, but tracking birds has been in place now for several years, and they're starting to learn things about, for example, the avian flu spread, through the connections between migration routes that they did not know.
Yes, thank you for talking. I always wondered why animals are so social. Like human, I was wondering if you found some similarities between social networking and humans. I was also wondering this study if the result can be applied to something practical, such as, saving species or some other application.
What species?
I mean, like a study, do you have any other application? Is just a part of knowing why they are social? And
Yes.
what is the greater objective?
Right. So in general, I mean, I use wildlife social as an example, but for the two species we're talking about, the big question there is actually the conservation. This is species that are very, very different. Well, ecology in general, sort of what is the difference of their ecology, but with such similarity in the two species and the similarity in their ranges, home ranges and habitat, why is one thriving, the other one is nearly extinct? It's a conservation question and a sort of deep ecological question. But understanding for animals, we sort of work with different species. The tools that we develop are not specific to zebras or baboons. In fact, the same. And we have a whole suite of dynamic network analysis tools that we've now developed over the last 10 years. This particular dynamic communities is this general dynamic clustering tool, which is now we're using to analyze brain activity and brain dynamics of brain networks in collaboration with Tanya from also the University of Urbana-Champaign, to understanding the patterns of at the cellular level. So the images there from brain imaging at the level of slices and live brains, where each pixel corresponds to about 4 cells in the brain, or neurons. So we can really start understanding the dynamic of the brain, and we can understand the clustering of functionality of those cells. And that's we're using the same tool. Humans for sure. We sort of, again, the tools at the level of abstraction that we develop them do not care whether it's horses, zebras, or baboons, or humans, or brain cells. But, and so it's a question of application. I see sort of the big bulk of my focus is developing the tools, the computational tools, and the applications that I'm interested in are certainly in animal biology, but they're applicable to in any domain where the questions are about dynamics of networks, of interaction networks. Now, in terms of similarity between humans and baboons, we have a sister project led by Dr. Suni Lemon from Technical University of Denmark, where they gave smartphones to several hundred, now coming up to 1,000 of undergraduate students at Technical University of Denmark. And they're tracing their social network very, very similar to sort of our baboon social network. So we're really hoping that, and I was going to Denmark in the fall to apply methods that we developed for baboon social network analysis. And for baboons, the question there is decision-making in a social species. So these baboons, they wake up, as you saw in that little movie, One Day in the Life of a Baboon. I can show a lot more data. They wake up from these trees, they go down, they eat, and you can see sort of the groupings, and then they suddenly decide to move, and you see them single file going to the next location, and they stop and they eat, forage, spread out, come back together, move. So these decisions of stop and eat versus, okay, get up and move to the next location are somehow propagated through the whole population, just like our decisions in the human population. There's the propagation of influence, propagation of behavior. These are the same questions that we're asking. How is the decision made in the social context, in the context of a species which has some hierarchical structure and has some social structure to move? Can we predict using their location using the social interaction? And can we predict can we extract social interactions purely from their location data? So that translates directly to human context, and we're hoping to apply that to sort of the data from the cell phones. But it also has huge implications to privacy in humans. I gave a version of sort of more oriented towards location and social network of animals as mobile social users. Because if you can purely from location data and past history of location data of a group of individuals, predict the location of each and another individual, leveraging that social structure, how much does it tell you about that device you're carrying with you? The privacy of that device, and the privacy of the data that comes to all the apps that get it out of it. Do you want to disclose your location privacy? And if you don't, how much of it can be extracted from the locations of those with whom you interact? Any questions? Okay. I'll be around also to answer questions in a more one-on-one interaction, social interaction. So thank you very much.